Okay, so previously on Earth Final Conflict, we have stabilizing eye movements. So last time, last time we did looking at the same thing, and so we had vestibulo-ocular reflex and optokinetic nystagmus, and then pursuit, which was kind of like a version of optokinetic nystagmus, where you just look at one thing, a small thing. So now, what we want to do today is look at something new. So when we're looking at something new, we do that all the time. I mean, like, typically a couple times a second, you're moving around and you're just like, checking out new things. And so to get your fovea, the highest resolution part of your retina, to go to a particular place. So when you first hear about this, you might think, well, it's kind of a low-level thing. It's just like involved in sort of like moving your eyes around. But a large chunk of your brain is doing this. So we're talking about the superior colliculus today a lot, but it turns out the signals that come through the superior colliculus go to all different parts of frontal cortex and parietal cortex and are heavily used for sort of getting around in the world. So this is actually quite it's a thing of many levels. But let's just start off with kind of the very most basic circuit for doing this orienting movement, which is the circuit in the superior colliculus that gets a retinal input and then moves the eyes so that the center of gaze gets to a particular object. So what are the main parts? Well, we've got the eyeball. So there's the eyeball, and then here's a muscle. There's an eye muscle. So and so what are our boxes? So our boxes here, we've got the first box is the superficial colliculus. So what is the colliculus? It's kind of a layered structure, like a layer cake, kind of like the cortex. So this is the superficial superior colliculus. Colliculus just means little hill. Colliculus, because the inferior colliculus is the inferior little hill, so it's actually a little mound. And so it's a complicated structure. It's got like about 50 different kinds of cells at each point in the colliculus, kind of like the retina. Like if you think of the retina, you got X cells and Y cells and on cells and green cells and blue cells, just like you got amacrine cells, horizontal cells, tons of stuff in there. Same thing in the superior colliculus. We're going to summarize it down to just two things, maybe three things, but into initially the superficial colliculus and the deep superior colliculus. Superior colliculus. So we got superficial and deep superior colliculus. And the cells in the retina come in and project to the superficial. There's a visual map in the superficial superior colliculus, and that goes down to the deep superior colliculus. And then what happens is it goes down to some structures down in the brainstem that have absolutely ridiculous names, and so we'll call them H for horizontal and V for vertical. So, and I'll go over these in more detail. So the horizontal is the paramedian pontine reticular formation, and the vertical is the rostral interstitial nucleus of the medial longitudinal fasciculus. But we know them as horizontal and vertical. And so what happens is the brain is kind of Cartesian, so it just sort of works things out. We've got some specialized circuitry that just concerns horizontal component of eye movements, and then another specialized circuitry that just controls the vertical component. And the superior colliculus goes to both of them, because obviously it needs to control both horizontal and vertical. And then those structures project to the oculomotor nuclei that we knew about before. That was the 12 oculomotor. So there's 12 of these guys, 6 for each eye. And then that goes out. So these guys then project down here. And then this guy finally goes back and synapses on the eye muscle. So remember, this system is all, it's running during all of these things. So you might be doing, vestibulo-ocular reflex, like you're trying to look at this straight ahead and you're moving your head around like this, and the vestibulo-ocular reflex is doing its job and keeping your eyes focused on whatever you're looking at here. And then you might decide in the middle of that head movement, I'm going to look at something else over here. So this system somehow has to, integrate with this system. So that same system that is driving the eye muscles around suddenly will have another signal superimposed on it, because there were all these other things coming down here, like the vestibular nuclei, right? It's hard to sort of keep everything in mind. There's like about 70 parts actually. So the vestibular nuclei are just doing their thing and moving the eyes around, and then in addition you've got these saccade nuclei, and so these are, we call these, the saccade. Saccade is just a rapid eye movement. Saccade pattern generators, and we'll talk about that more when we talk about like walking or repetitive structured movements. So pattern generators. So these mainly generate the burst. So they generate the little burst that sort of accelerates your eyes to move to a new place. So those are saccade pattern generators, H and V, just for short. Now, it turns out the rest of the brains is heavily involved in this, and so if I draw another box, I'm drawing it at the same level as the superficial superior colliculus, but that's the visual cortex, a bunch of areas in the visual cortex. Pretty much almost every area in the visual cortex, including the highest ones, project to the superior colliculus. So there's a lot of areas, like we're talking 30 or 40 areas are going down to the superior colliculus. And what's the equivalent of the deep superior colliculus? It's something called the frontal eye fields. And so why did I draw it at the same level as the deep superior colliculus, which is just a piece of the colliculus? The reason I did is because this guy has parallel access to these pattern generators. So neurons here go all the way down to the brainstem to H and V, and they actually have parallel control of those neurons. And similarly, if you look at the retina goes over here to visual cortex. There's a bunch of connections there that I left out, 30 areas. But visual cortical areas project to the frontal eye fields. And so you've got two parallel systems for controlling eye movements, and they can both interact. And if you actually stimulate both of them, you'll actually get the vector sum. Like if you stimulate this one and you get an eye movement off to the left, and you stimulate this one and you get an eye movement that goes straight up, stimulate them both at the same time, you get a combined eye movement. So they really are just parallel control mechanisms. Now they've got to somehow, talk to each other. So, visual cortex, so the frontal eye fields also go to the deep superior colliculus to tell, like, the deep superior colliculus, I'm going to do something. So maybe don't do something because I'm doing it, or maybe do it in addition, whatever. They got to sort of communicate with each other. So there's a lot of extra connections, but these are our basic sort of final common output pathway connections. So you got the superficial colliculus projects to the deep, visual cortex, visual cortical areas, project, a lot of these go here, and then these go here. But if we sort of just look at our original diagram, you got visual things, motor map, visual things, motor map, And then both motor maps talk to H and V in order to make the eye movement. So if we now sort of look in a little more detail at the superior colliculus, so let's divide it into a couple, maybe one extra area. So like I said, there's at least 50 cell types in here. And what does that mean? It means that at each point in the superior colliculus, there's literally 50 different kinds of cells. Long ones, big ones, ones that go into this layer, ones that go to that layer. It's a complicated structure. But we're going to summarize it down to just 3 layers. So we'll make them like little plates. So here's one layer. We'll call that the superficial one. And then we'll make another one that's underneath that. That's kind of the intermediate one. And then another one under that. And that's the deep one. I just went off into the ozone there. Okay, so we just have superficial, intermediate, and deep. And so this initial things that I'm going to tell you about as far as how the colliculus works were figured out a long time ago, like 70 years ago. They already knew this 70 years ago. So that's one of the things that I say about neuroscience. It's good because the brain doesn't change. Nothing has changed from 70 years ago. Same brain. That's good. So basically, there's several different things you can do. You can record from the colliculus, say the superficial or the deep, and then you can stimulate in the colliculus, superficial or deep, and then just see what happens. And so what are those things? So we can basically record the receptive field, like find out what part of the visual field that part of the colliculus is representing. Or we can record during an eye movement. So we can have train an animal to move its eyes around and just find out what's going on in the colliculus. Or we can electrically stimulate to find out what happens to an eye movement if you stimulate. So those are 3 different kinds of experiments. And what are the conclusions from these experiments? It was basically that we have a retinal map in the superficial colliculus, and then we've got something a little more complicated going on in the intermediate. We'll just leave that out for now. And then we've got a motor map down here. So what does that mean? So like I said, 50 years ago, they knew there was a motor map in the deep colliculus. And so what those 3 kinds of experiments are, to see why people thought that was a motor map, if you look at, so this is the visual field I'm drawing here. So we look at the visual field. So the animal's fixating there. So the first thing we might want to do is just have the animal. So obviously, in order to do these kinds of experiments, you have to have the animals control where it's looking. So we have the animal just fixate at the center, and then just record. Go into the superficial colliculus, just record. And what we'll find is say a receptive field out here, something like that. So this is receptive field. I'll write it out. Receptive field, superficial collic. superficial, write that out. Superior, that sup means superior. Superficial, so receptive field in the superficial colliculus. And if we go someplace else in the colliculus, you find them. You'll find the receptive field. You know, there's a visual map. The receptive field will grow someplace else. So now what we can do is record. This is a little more complicated. Record. Go to that place and record during an eye movement. And if we record from an eye movement, so here's where we're recording. So now let's go down, say, into the intermediate or deep superior colliculus, and record. So let's record down here during an eye movement. And so that's a little more complicated because you have to have the animal, you know, move its eyes all over the place. So this guy goes up here. So now let's record down here and have the animal make all different eye movements. Just, you know, train the animal to look left, look up, look right but not as far, look down, not as far. You have to have an animal look all over the visual field. And what you'll find is we'll find a movement field. It's a little bigger. So I'll make that one in dark. So that's the receptive field. But now we're recording a movement field. And what is the movement field? Movement field. So this is the receptive field. This is the movement field. And that's basically what movements does the deep part of the colliculus become active for? And not surprisingly, it's active for movements that are sort of in this general direction. So what do those movements look like? That means, like, if the animal makes a saccade like that, it would respond. If it makes a saccade like that, it would respond. If it makes a saccade like that, it would respond. So this whole family of saccades, whenever the animal makes one of those saccades, this motor map responds. And so that's basically how you know it's a motor map. And a more brute force method is, why don't we just stimulate down there? So what we could do is, instead, we could just actually put some current through the electrode. So instead of, make that one a blue electrode, so stim, we could just put some current at that point. And what will happen is, you'll get this saccade. So there's this guy's, I'll make him nice and big. So it's kind of hard to tell the difference between blue and green there. Let's make him a red. No, I already had a red. Okay, well, let's just erase that guy so we can see some blue. Okay, so here's what you get out of stimulating that part of the colliculus. And so you can see that all these methods together show you that you've got a sensory map up here, a retinal map, and then you've got a motor map down here. So this motor map basically is arranged so that when you stimulate the motor map, immediately underneath a retinal map that represents this part of the world, what you get is a saccade that will essentially move the eyes to that part of the world. So it makes sense to set it up this way. A target comes up here. If we stimulate this part of the colliculus down here, it's going to move the eyes to that target. And it's a motor map because, you know, as you stimulate in different parts, you get slightly different, you know, there's actually a systematic map of eye movements. So some parts of the colliculus represent small eye movements, other ones represent large ones, and different parts of the colliculus represent, you know, movements in different directions. So you actually just have like a map which is lined up with this retinal map in such a way that if we just take some activity from the retinal map, put it down in the motor map, it will make the eyes bring the center of gaze to where that peripheral receptive field was. So remember, the goal is the animal is just actually looking at this point right here, and our goal of the colliculus is, let's get the sensitive part that's looking at the center to look at the new thing that's off in the periphery. So once the animal looks at it, it's no longer in the periphery. Now it's back to the center, because that's the whole goal of it, is try to get the sensitive center to look at something that's interesting off in the periphery. Okay, so that seems reasonable enough, easy to understand, reasonably easy to understand. But it turns out it's more subtle than this. So before I go on, is everybody okay with the basic sensory map? So we got a retinal map here, we got a motor map of different saccade vectors. That means there's a saccade has a certain direction and it's got a certain length. So it's a two-dimensional map of these saccade vectors, and they're all lined up perfectly so that whenever we stimulate the deep part that's underneath a particular map location, it will cause the center of gaze to go to that map location. Good so far. Okay, so now all hell breaks loose. So it turns out there's a problem that comes up. So there's all this junk out there, and it varies in how interesting it is. So sometimes, like if you're an animal, there's a leaf fluttering around, causes the superficial superior colliculus to become active. Who cares? It's just a leaf. And then there's just like a little predatory bird. If you're a small bird, like a sparrow, you see this hawk off in the distance. It's just a little tiny little hawk-like movement. Now that's a really important location to pay attention to. But maybe the hawk was hiding in a tree and it stopped moving. You still need to pay attention to that location. And so really, what the colliculus does, it's not just a retinal map over a motor map. It's a map of interesting things to look at. And so people have called it, instead of just a retinal map over a motor map, they've called it a saliency map. So you've got retinal things. That's just like, the retina is responding because something is in a particular place. But then you have saliency. Is this an important part of the map? And then you got the motor map. So you can sort of think of those, along a continuum. So you start off with retinal, that's just, is something occurring there. But then you've got this other thing, which is, how interesting is this part of the world? That's a different thing than something it might be an interesting part of the world, and maybe the predator has stopped moving and you can't actually see it anymore, but it's still interesting. And then you got the motor map. That's actually what you're going to look at. Okay, so what is one of the key experiments that people did to show that something a little more subtle is going on in the colliculus? And this was an influential experiment by Hallett and Lightstone. If you want to go back and read this. Lightstone, 1976. See, a long time ago. Good experiment. And so what they did was, it was a human experiment. They just had people, they basically just turned on lights for briefly, so this is time, and they just turned on lights briefly in rapid succession. So there's light 1, light 2, light 3, light 4, light 5. So just turned on lights. And so here's where they are in the world. So say here's 1, and then there's 2, and 3, and 4, and 5. Now, they're not all on at the same time. They're just one at a time. But if you turn them on slow, like, light, 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 then it's no problem. You just look at light 1 comes on. So here's the center. So light 1 comes on, and so you look at light 1. And then light 2 comes on, look at light 2. And then 3, look at light 3. And then 4, look at light 4, 5. Okay, so we're all good. except I managed to overlap 4 with the center. Let's try that one more time. There's light 1, and here's the center. So the very first thing is we look from the center to light 1, and then 1 to 2, 2 to 3, 3 to 4, and then 4 to 5. Okay, now we got them all unoverlapped. Okay, so that's a very straightforward thing, no problem. You can do it very easily. Now all they did was they just sped it up. So if you speed it up, so now let's just do it, speed it way up. And this is an effortless thing to do. So that's the important point. It's not like, it's not effortful. You just say, you just tell the person, just look at the lights. You know, whenever they come up, look at them. Now if you go really fast, so if you go slow, like, you know, light 1, 2, 3, 4, 5, you can easily do it. But if you go really fast, like da-da-da-da-da-da-da-da-da-da, you can still do it. But what's actually going on when you do that? It's interesting, because if you monitor the people's eye movements, what happens is, first you turn on light 1, they're still fixating the center, and then it goes off. Then you turn on light 2, they're still fixating the center, then it goes off. Then you turn on light 3, they're still fixating the center, getting ready to look at 1, it goes off. Okay, now there's nothing on the screen, and they make an eye movement to where 1 used to be. So that's where 1 used to be. It's no longer there, long gone. And then what happens is light 4 comes on, and maybe even while light 4 is on, they're now making a saccade to where 2 used to be. And then, you know, light 4 goes off, and then light 5 comes on. Now they make a saccade to, you know, where 3 used to be. Now, if you were following along, it's going to be a problem for this system. Because what does this system do? This system basically aligns up a particular saccade with a particular receptive field. But what's going on here? The stimuli are long gone, and your eyes have gone all over the place. So once people did this experiment and realized it's trivial to do this experiment, people can easily do this. They can get 2 or 3 behind, no problem at all. And they can continue looking at where the previous locations were. It was clear that there's got to be some kind of updating mechanism. So these were psychologists originally doing this, psychophysicists doing this, but they realized there had to be some kind of updating mechanism that was dealing with this simple sensory map over motor map. A simple sensory map over motor map's not going to cut it when you've got like light 5 coming on while you're making a saccade from light 2 to light 3. And so basically, people did a bunch of experiments, and they generally fall under the category of, if you want to read about these, double-step saccade. And we'll go over one experiment to sort of try to understand how this works. Now, in the end, it will be all good and be sort of easy to understand, I'm hoping. But let's just look at what's going on when we do a very simple experiment where we put up 2 lights, one after the other, before the animal has had a chance to move its eyes. And then we just ask the animal to look at the first one, where the first one was, and then look at where the second one was, and just see what goes on in the colliculus during this. And this will hopefully make it clear how you could do something like this, how you could somehow, you know, buffer these targets. Because what's happens is, like, something happens and then it goes away. Like a predator comes and then it doesn't move. But you got to remember where it was. Yeah.
Okay, just to summarize, make sure I'm understanding correctly. So, is it like the saccade, the second box picture? Is that the empty one.
Where it was, yeah, where the object was.
So is that like is that like the retinal map or like the motor area kind of work ahead?
Yeah, I mean, so the problem is there's several things going on. You have this retinal map, but it's movable. And so now the problem is we have to sort of, so when the person is fixating, they've got the retinal map sort of with the center on the fixation point. As soon as they make a saccade to where one used to be, they move their retinal map to this new location. And so you can see that there's going to be a problem if we just access the motor map. It's not going to give us the right saccade because we removed the retinal map. I'll draw the whole thing out in detail just for 2, so we can sort of see what's going on. Hopefully that will make it clear. This should just give you a feeling like something fishy is going on, because you can actually saccade. You can get like 3 behind, so somebody's got to be keeping track of where all those other targets are. Okay, so we have 1, 2, 3, 4, 5, 6. So we got 6 boxes now. So we first have world view. So this is sort of world view and colliculus view. So this is world view up here, and so we've got six boxes, and we'll just draw out 6 time points in the world. So, and then we'll have 6 time points in the colliculus for just a double step. 1, 2, 3, 4, 5. Okay, so we got 6 time points, and so this is the visual world here. There's the center of the world. It's right there. So you can just think of this as like a screen that the monkey is looking at. So monkey is looking at that screen. And so what's the goal? So what's the overall goal is I'll just put it over here. We turn on a light at 1, and then we turn on a light at 2. Then the lights are both off, and then the animal makes a saccade to where 1 used to be, and then it makes a saccade to where 2 used to be. So that's so this is stim, and then the stim is off, and this is the response. So this is, he's making a saccade to where those lights used to be. They're turned off now. So this is double step. This is the basic double step idea. So basically we turn on light 1, turn it off, turn on light 2, turn it off. Animal's still looking at the center. Animal's still looking at the center. And then in the dark, it just saccades to where 1 used to be, and then where 2 used to be. And so if we draw that out on the on the world here, so first the animal's looking at the center, then it's still looking at the center. That's what the X is supposed to mean. And a light comes on at 1, and it's still looking at the center, and a light comes on at 2. Then the lights are off. It's just looking at the center. Now it makes a saccade to where 1 used to be, so that's where 1 used to be, and then it makes a saccade to where 2 used to be. So very simple. Should be just like, seems like a straightforward thing. And it's trivial to do. Like if you just do this, just put up 2 lights, and you can look at where 1 used to be, where the other one used to be. It's not a problem. I mean, it's not hard. So now let's look at the colliculus. And so the colliculus has 2 halves. Remember, one half is representing, like the right colliculus is representing the left half of the world, and the left colliculus is representing the right half of the world. Let's just forget that for time being. I'll just draw the colliculus kind of lined up with the world so that it doesn't sort of confuse us too much. So this is superior colliculus. Okay, so now we're in the superior colliculus. And like I said, I'm going to sort of cheat and just say, here's the center of gaze of the colliculus. Like we'll put that little star in there as the center of gaze of the colliculus. And so it's kind of a round thing. And so here's in real life, the left part of the visual field is dealt with by the right colliculus, but let's just pretend for sake of illustration that the right colliculus is lined up with the right visual field. So this is, right visual field. So that I don't have to mirror image everything. It makes it a little clearer. Okay, so now we got a bunch of colliculuses. So let's make our colliculi down here. There's the center. Kind of get a little shrunken. Okay, so here's the center. Got six colliculi. Okay. Another one. And the last one. Okay, 6 colliculi. So this is time going this way, time's going that way. So what happens? So what happens at the beginning? Well, there's a little activity at the center of the colliculus. So I'll draw it in. Did that in green. Okay, so a little activity in the center of the colliculus because the animal's fixating. And now there's a little activity at 2 places in the colliculus because the animal's fixating there, but then target 1 comes on. So then the animal's still fixating and target 2 comes on. So these are superficial collicular responses. So this is superficial. So that's the retinal map. So it responds to those. Okay, so now all the lights go off, and so there's still a little bit of activity in the center. So now the animal's getting ready to look at 1. It's getting ready to make a saccade to 1. And so what happens at this point, when it's getting ready to make a saccade at 1, some deep colliculus activity comes on here. So this is deep. Because we know that we've got a retinal map on top of a motor map. So this is deep colliculus, and what does that part of the deep colliculus do if you stimulate it? It makes a horizontal eye movement so the center of gaze gets over that point in the visual field. So it just does what we thought of at the beginning. We got a sensory map, a retinal map, and then a motor map. And the motor map is lined up so that if something stimulates at this part on the sensory map, if we go down and stimulate that part of the motor map, it just makes a saccade so that we get whatever is out there in our center of gaze. So that's all good. So some activity appears there, and then eventually that activity gets loud, and we get a saccade. So the animal makes that saccade. Okay, so now what happens? Well, this activity will have gone back to the center, right? Because there's nothing on the screen, but if there was something on the screen, say target had stayed on, say target 1 had stayed on. If it had stayed on, we would be looking at it. And so there would actually be some activity there, but there isn't because the screen's black. Yeah.
Do you mind rewinding a little bit?
No problem.
How do you identify which is a retinal map and a motor map when looking at the circle?
Okay, yeah. So we're looking down. So now, unfortunately, yes, we're looking down on the colliculus. So here's the layer cake of the colliculus, and so here's the. So here's the retinal map, And then here's the motor map underneath it. Yeah, so that's an important point. Yeah, look at looking down. So here was our original, motor map over sensory map like this. So we're looking down on it like that. Yeah, that's a good question. Yeah, I should have said that. Yeah. So yeah.
Is it like a horizontal and like vertical thing? Okay, if you identify the little area that's in the deep colliculus, is that in the motor map?
Yes, that's in the motor map. Yeah, deep colliculus. Yeah, deep colliculus activity in motor map.
And why is that the motor map? I'm so sorry.
No, it's no problem. No, no. So it's the motor map because if we put an electrode in the deep part of the colliculus, if we just go down into that deep part of the colliculus like that and put an electrode in there and stimulate, it makes an eye movement, a particular eye movement. So if we went to that part of the superior colliculus and stimulated, we would get a horizontal eye movement. If you stimulate it again, you get another horizontal eye movement.
You're going deeper in there because you're going to the deep colliculus, but it's in the motor.
Yeah, it's in the motor map. So if you stimulate in the superficial map, the animal might see something, but it won't necessarily make an eye movement. If you go into the deep map, it'll actually make an eye movement.
Okay, and then the superficial is in the retinal map, it's at the top.
It's at the top, yeah, and it just responds basically when something is actually out there.
And that's the retinal map.
That's the retinal map.
Okay. So it's a process to get like farther and farther down there, the circles are really deeper and deeper.
Yeah, so normally, so everything's great as long as we haven't made a second saccade. So for the first saccade, there's no problem. Like, so if something comes up in this part of the superficial map, all we got to do is go down to that part of the deep map, turn it on, and we get the right saccade.
So it's like a card with a restart, kind of we're looking at something different, and I have to readjust everything.
Yeah, it's basically just move the eyeball. The saccade is move the eyeball so that the center of gaze, your fovea, gets onto whatever was out there. So it's just like you're looking straight ahead, something really interesting happens up here. That part of the retinal map is over a part of the motor map that if you stimulated it would cause you to look at that thing. Okay, so that's all good, except now we've got a second saccade. And so now what happens? So if we just went to the part of the motor map that's underneath number 2, that would be here. So say we went to this part of the motor map and stimulated it. So this is motor map location under number 2. So say we just went there and stimulated that. What would happen? What would happen is we would get a saccade. And what saccade would that be? Well, it would be a 45-degree angle saccade, right? So like when we stimulated this part of the motor map, we got a horizontal saccade. If we stimulate that part, we're going to get a sort of an upward 45-degree angle saccade. And so what would that look like? Well, the animal is now looking at this part of the world, and what would happen is we would get a 45-degree angle saccade like that. Let's make that one dashed, because that's what would happen if we stimulated that part. And that's not right, because it hasn't taken into consideration what you just looked at when you looked at 1. So now you can see what the problem is with the motor map and the retinal map. If it was just a fixed motor map, it wouldn't work for this setup, because it would work for the saccade to 1, but then the problem is when we make a saccade to 2, we're not starting from the center, so we don't need a 45-degree angle saccade, we just need a vertical saccade, because we have to take into consideration, we already went to 1, and so we're going from 1 to 2, not from 0 to 2. So so what happens? Well, when people first did this, they said, well, all we've got to do is we've got to do a vector subtraction of the first saccade. So what is that? Well, if you take this saccade, take that saccade, and subtract it off of this one. So subtract it off of this one means like take that saccade and flip the vector and subtract it off that one, and then we're done. Okay?
So basically the center gaze is the number 1, and when you simulate the motor movement, instead of going to number 2, which would be the thought, it's doing the movement itself of 45 degrees. It's not necessarily the point fixation that we're looking at, it's the angle that we're concentrating on.
No, there's one mistake there, which was that the center of gaze is the X, and 1 is the first thing you're going to look at, and 2 is the second thing you're going to look at.
Yes, but from what I'm understanding, so the last picture is the center gaze became, when you stimulated number 2 in the motor movement, the center gaze toward the monkey was 1 instead of it being the center gaze.
Yes, right, yeah. So the center of gaze at this point, exactly. So at this point, this is, center of gaze here.
And that's the problem.
Yeah, that's the problem, because the center gaze is moved, and this first activity in the retinal map is appropriate to the center of gaze being at the center, but it's no longer at the center. So we somehow have to sort of update things. And so what happens is that part of the deep colliculus does not become active. What instead happens is it somehow gets shifted over to the right place. And where's the right place? The right place is over here. Sorry, I'm drawing it on the visual field, not the deep colliculus. Sorry. So here's, okay, so let's go down to the colliculus. So if we turned on this part of the colliculus, we would get a 45-degree saccade. We would get this one. But we don't want that one. What we want is a vertical one. And so how does it figure out how to make a vertical one? Well, what it does is it basically doesn't do that one. Instead, it subtracts off the first saccade and moves the deep layer activity to the appropriate place. And then when that part of the colliculus becomes strongly active, it causes the correct saccade. So it actually causes this correct vertical saccade that happens.
Sorry, just to clarify.
No, it's good.
I was like trying to wrap my head when you're saying like saccades 1 and 2.
Yeah.
Does that mean like use the example like saccade 1, like you're focusing on the top, right? Would saccade 2 be like a completely different object of like focus, or is it like if it's moved? Because like how you said if it moves, like it changes. So like when you have like more than one saccade, does that mean like the object you're looking at has like moved or moved, or just like?
Well, so remember here, the two objects are off. So the animal is looking at the center. An object comes on, goes off. Another object comes on, goes off. Now there's nothing on. Now it looks at where object 1 used to be, and then it looks at where object 2 used to be.
Okay. Yeah. Oh, wait. Okay. Hold on a second here. Is it because, so, you said the like little 45-degree angle thing, is it because it's not moving like straightly with,
No, it's not because of that. No, the problem is, the problem is that you've got to somehow take into consideration where you went to, because you moved your center of gaze to where object 1 was. If you hadn't moved your center of gaze to where object 1 was, and you were still looking at the actual center, then the 45 would have been correct. But we somehow have to sort of subtract out the effect of where we looked. And so when you first think of this, it just seems like, it's mind-bogglingly complicated. It's not. It's actually very... No, it's straightforward. So here's how it's straightforward. So imagine this is what the colliculus does every day. So that's the world, and here's the colliculus. So this is just a day in the life of the colliculus. So what does the colliculus do every day? Well, there's junk out in the world. So let's just say there's like this thing out in the world. Okay, there's a big thing like that out in the world. And say we look at some part of it. So say we look at, let me make it a little bit off to the side. So here's like a triangle out in the world, and we look at, say we decide to look at this part of the triangle. So what's going to happen in the colliculus? So now that's the world. So this is, remember, this is world, and this is superior colliculus. So what's going to happen in the colliculus? Well, before we've looked at something, you're going to see that object is going to be in the colliculus like this. Okay, there it is in the colliculus. Now we have to decide what to look at. So let's say we look at this guy. So say we essentially like make a saccade to that guy. So this thing is off in our periphery, and then we just look at the bottom of the triangle. What's going to happen in the colliculus? Well, whatever you're looking at is going to end up in the center because you looked at it.
Oh, so it's a new center of focus?
Yeah, it's a new center of focus. And so then where's the rest of it going to be? Well, it's going to be like this. What about this guy? This guy is going to go into the other colliculus. So that triangle is going to move down like that. So that's, you just looked at one thing. And what happened was, whatever you looked at, everything else is going to move parallel to it. So whatever you looked at, so that's what you looked at. So this guy is just going to, I didn't draw it very accurately. So basically everything is going to move parallel to whatever you looked at. So this guy's going to go here. This guy's going to go here. And so essentially what's going to happen is the object is just going to rotate over the surface of the colliculus, like I describe it like a planetarium. And everything is going to move parallel to whatever you looked at. So, there's a whole complicated world out there. And if I just look at this part, say I'm looking right here, and then there's like this complicated thing out there, and I look at that thing. What happens is this whole thing in the colliculus just all moves parallel to whatever I looked at. It just shifts on the colliculus. Parallel to the line I looked at. So you can just sort of think of like if you could see the world on the surface of the colliculus when you looked at something, what's going to happen? The whole world is just going to shift on the colliculus, and it will shift parallel to whatever you looked at. Well, yeah, there's a good. That's another question. Like, why does if the world's like every quarter of a second the world's shifting around the colliculus, so it seems like everything should be moving. Yes, but this whole system is really wired into your whole higher level cortex so that it knows, oh, it's about to shift, and then it shifts every quarter of a second, and then your brain says, oh, okay, that's what I was expecting. Yeah. Yeah, your brain, the colliculus sends all this information up to the brain and says, like, this is what we're doing. And then the higher brain says, oh, okay, yeah, I was expecting that.
I have a good question, just to make it, like, really real. So if I was looking at the chair,
yeah,
and then I'm like, it's fixated.
You're,
Like my little,
that's the cross, right? You're looking at the chair.
I go, man, I look over there. The whole thing just shifted in my periphery.
Yeah, that's right, exactly. Yeah, the chair. Yeah, that's exactly what would happen. So, yeah, so the chair now, if you're looking over to the left, the chair is just in your periphery. The whole thing just shifted. And so,
And that's what you fixated on area, because now I'm like,
yeah, because now you're looking at whatever's over there. Yeah, so basically what happens in the colliculus is just like every time you look at something, everything just like rotates over the colliculus. And then you look at this, goes here, goes here. It's just like constantly shifting around. Yeah. Yeah, it's just shifting around. That's what's happening, exactly. All the objects are shifting around. And so finally, here's the final insult, the final point. So how does that help us with this? So how it helps us with that is that what the colliculus does is it basically hallucinates where all targets would have moved to.
Is this part of the brain making up what your eye doesn't necessarily concentrate on?
Yes, no, exactly. Yes, this is kind of like, it's a map of salient things. So it hallucinates where all the targets would have moved to if they had stayed on. So they got turned off. So this is what the colliculus does. So what it does is it says, this is what we see all day long. We see stuff shifting over the colliculus like that. But now, what if some of the targets go off? They're no longer on. They're no longer stimulating the superior colliculus. What the colliculus says is, that's okay. What I'm going to do is I'm going to shift where that target used to be to where it would have ended up if it had stayed on.
Okay, two things. One, so let me ask you this. When you say go off, do you mean like darkness?
Yeah, just darkness. Yeah, so like this, you turn on a light here, and then over here it's off.
But then it's on in number two.
Yeah, and then you turn a light on in number 2, and now all the lights are off. It's just pitch black.
And they look at number one, even though the lights are off.
Yeah, lights are off. You can do this trivially. It doesn't even seem like you're doing any work. Yeah, lights are completely off, and the animal just says, I'll look at where 2 used to be, and now I'll look at where 1 used to be, now I'll look at where 2 used to be.
And that's what they're fixated on at first, so they're just going to immediately look back to where they're
Yeah, to where the target used to be.
When you refer to the motor movement, is it just a matter of like the angular movement, or what was the problem with the motor movement?
So the problem with the motor movement is that so here's target 2, that causes superficial activity here. If we go to the deep part of this, the deep colliculus, and stimulate it, we'll get a 45-degree saccade. And so somehow we had to have every time we make a saccade, we've got to go back into the colliculus and move all the targets. Now, normally if they stay on, we don't have to move them because, they'll actually just move over the colliculus and they'll still be on, and they'll be on in the correct place. But what if the target went off? That's where you've got to hallucinate where they went to. So we got to that's what the colliculus is doing. It's kind of like, there's a tiger over here, big predator. Now it's stopped moving and you can't see it anymore. Now we make a saccade. What am I going to do? I'm going to essentially update things in retinal coordinates as to where the target would have been after I made the eye movement, even though I can't see it anymore. Yeah, that's the 45 degree. So because we made that first saccade, this is where the tiger used to be. We don't want to activate this deep part of the colliculus. We got to somehow this part of the colliculus. We have to somehow update it with our eye movement. So the eye movement information has to come back into the system and move the tiger target to the right place.
So you see the tiger, and then you
then it runs away, or it just stops moving. Yeah.
and you're like, oh shoot, I don't know where it went, and I think it went that way, and that's the 45-degree shift, because I think it's based off of where it's been before, I assume it's going to go this way, so I'm going to look in that direction. Is that the 45-degree thing?
Here's the thing: is so the target, the tiger shows up, and then the tiger goes off, like it stops moving. Now I look somewhere. And now I want to look at where the tiger used to be. That's the problem, because I had a saccade in between. If I just immediately look at where the tiger used to be, no problem. But what if I'm just looking at a bunch of stuff like this, and then I see a tiger, and I look over here, and now the tiger is invisible, so I can't use my retinal map to figure out where the tiger is. I have to use my updated location of where the tiger would have gone to if it had stayed on. That's the hallucinate.
Stay on is just like a word
If you could have seen it, right, exactly. Yeah, so that's what the colliculus is actually doing. It's not just retinal. What it's doing is it's updating a set of interesting to look at targets, whether or not they're actually visible or interesting at a particular time. So it's effortlessly sort of shifting around these targets, which is kind of like what happens to the colliculus normally, because stuff's just sort of shifting over the colliculus. So all it has to do is kind of shift this invisible target location. So that's why when you look at the colliculus, like the top part is just retinal. As you get to the middle layers, they're kind of more cognitive because they're the ones that are kind of shifting, shifting the salient. So this is like the saliency map is kind of in the middle layers, and then you got the really motor map, completely motor map down here. So you start off with, this is stuff that's actually out there. This is stuff that's lined up with the stuff that's out there, but it's maybe no longer visible, but it's interesting. And then here's the thing that actually does the motor movement. Yeah.
That's all it is, though. That's what you're saying, right? The motor movement is simply the movement with an angle. You would expect it to be in the number 2 area, but it's not because the number 2 area has moved parallel.
Yeah, because your center of gaze is now looking at that point, and so we don't want a 45, we want an up.
And that's why you did the vector. So you just found out. Okay. I don't want to. Okay, I got it.
No, this is a tough one, but it's important because you really see that the colliculus is not just like it's not a straightforward motor map. It's buffering interesting things to look at and updating them all the time, and it's all connected up to parietal cortex and frontal cortex, and it's the reason why the world doesn't jerk around, even though it is jerking around. V1 and colliculus, the world's just, every time you look at something, the world's just like jerking all over the place, but it all seems still because the colliculus has told everybody, this is what I've done. And this is the eye movement I made. So feed that back in and update all your maps so that you're not surprised. It is, it is like this is often called a reflex, the orienting reflex. So, it's very much a reflex. Just something comes on, look at it. But it's a lot more complicated because like two things come on. First I look at one, then I look at the other.
Right. I was just thinking see it, think about how important it is to do the motor part.
Yeah, no, it's very much a reflex. You don't have to sort of think about these things. You're just constantly, but think of it, you're moving through the world, all different stuff's happening, and something might happen, like a little jiggle, and then it stops, but you still want to look at it. So the signal, the visual signal that made it interesting is no longer there. That's constantly happening. Things are getting all different interestingness at all times. And you have to sort of figure out, like, there's 3 interesting things there, and that guy lost interest, but I know from what it was that it's really interesting, so I should really maintain that guy. And this one has a big visual signal, but I don't care, because it's just a leaf.
It's the same like when you hear something that is like not in your visual field, like it's coming from behind you, and then you like want to see.
We'll get to that exact thing in a later later lecture. Yeah. we won't go on anymore. But this is the key point. Is basically you kind of what the colliculus does is it especially the middle layers it kind of hallucinates where stuff would have gone to if it had stayed on. And that's really the saliency map. It's really trying to sort of determine what's a good thing to look at. Okay, you probably didn't think like the colliculus, you thought it was down there, but it's actually doing like, all these cool things, and it's telling the whole rest of the brain about it. So we'll come back to that. This is definitely a hard one to get a hold of. It took me like months to sort of, when I was trying to read these papers, to try to figure out what's actually going on. So it's not straightforward. Hopefully.